going to continue with the next speaker, and that is Dr. Andrew Limper, who's a longtime colleague and friend. He's going to talk to you about drug development and IPF.
Laser. Thank you very much. I have to add my thanks and appreciation to Tim and Lisa, who have just put together a fantastic program. And I also have to thank Jesse Roman, because I think Jesse got me engaged in the Pulmonary Fibrosis Foundation really near the infancy of the PFF. And I belong to many academic organizations, but no organization is doing more for the real needs of our patients and putting forward the future than PFF. And if you go to these summits over time, which I've had the benefit of doing, we're on an upstroke that is on an exponential growth curve, and it's really, really exciting to see. So what I hope I could do now is to synopsis some of the stuff that is in the pipeline and some of the thinking about where the horizon is for drug development for this disease, and I think it gives hope. I tell my patients, I've been studying IPF and pulmonary fibrosis, taking care of patients, and very engaged for about 30 years now. And more has happened in the last 3 to 5 years than the prior 25 years. So I think there's a lot on the horizon to be hopeful for. So I don't have any relevant financial disclosures with respect to the contents of this presentation. What we're planning to do is review some of the recent and current trials of agents to treat pulmonary fibrosis. We're going to talk about the emerging concepts in the pathogenesis that are leading to the therapeutic targets. There's a whole host of new therapeutic targets. And we're going to discuss evolving thinking of drug targets in IPF and strategies for new drug therapeutic development. So I have been around IPF for a while. This is an old pathogenesis slide. It started out, let's see where the beamer's at, with some kind of an epithelial injury and activation. Didn't know what that was back in 2000. Have a host of inflammatory mediators, central roles for transforming growth factor beta and PDGF, TNF alpha, leading to things at that time. There was a lot of press and research on epithelial mesenchymal transformation. Myofibroblast formation, that's kind of fallen by the wayside, although there's still some evidence that it's important. And these formation of these pathogenic fibroblastic foci. If you look at 7 years later, we know a lot more about the details, but we really don't know what's going on. There are these nice lightning bolts that are zapping the lungs. So if you ask people then what zaps the lungs in IPF, it's always lightning bolts. But we know a lot more about a variety of cytokines, growth factors, gelatinase, metalloproteases, angiogenesis. Again, the myofibroblasts and the fibroblastic foci are important. And I look at this slide, and I think this is what we've been thinking about this, that we know a lot about these details of the process, but we have the something bad happens. I think within the last several years, we're starting to understand what the causes of idiopathic pulmonary fibrosis are in pulmonary fibrosis. This is a nice review by Mora, Annie Pardo, and Moises Selman. You got to pull this out, Nature's Review, just within the last number of months. And what we're really coming to the grips with, and you've got this here, is that we have the effects of aging, we have genetic predispositions, and we've talked a lot about telomerase, surfactant proteins, the MUC5B story, and then there are a host of environmental damaging agents. For some folks, it's smoking. In the Midwest, I have a lot of farmers who don't smoke, but they are exposed to agriculture exposures. They don't have hypersensitivity pneumonitis. They have UIP, IPF on the basis of having a genetic predisposition or tendency, and grain dust. And of course, a host of other things. Tim Blackwell was talking about herpes virus. So a whole host of other damaging things leads to this activation of this stress, endoplasmic reticulum stress response of the senescent epithelium. There is also evidence of senescent fibroblasts. There's a secretion of the senescence-associated secretory profile that actually helps to drive the fibroproliferative response. So when I was here at a few summits ago, Kevin Brown gave a talk like this. And so what Kevin Brown did was he went to clinicaltrials.gov and he pulled this out. And if you put IPF in, and I did this on a Saturday morning a month ago, you get 82 studies. So I said, oh boy, I'm going to have to put my talk together. I made a pot of coffee, about 3 gallons worth, and I sat down to kill myself to go through all the studies. I also put a beer in the refrigerator knowing I'd need to calm down after all that at the end. Well, I actually opened up that study from Moises Selman's group, or that review, I should say, and indeed, he did the work for me, or they did the work for me. So this is an outstanding article to pull. It has every target that is, most of the targets that are in PIPE that are being studied. They're arranged by those that are positive results, and of course, our launched agents, as you're familiar with, pirfenidone and nintedanib in blue. Negative results. We went through a whole host of those, studied over time, and you'll see, N-acetylcysteine, of course, that jury is still out, as we know, with the TOLIP genetic sequences. Interferon gamma, sildenafil, we'll get back to sildenafil, imatinib, triple therapy, prednisone, warfarin, bosentan, and ambrisentan, all being negative studies in red. There are a host of things in phase 2. What's nice about this review is that actually you can find any given agent, which oftentimes the pharma companies give them numeric and letter descriptions or monikers, and you can see that they have ongoing trials in green, such as pamrevlumab, which should be out, I think, within about 6 months, treprostinil for pulmonary hypertension in association with pulmonary fibrosis, a whole host of agents that have pending results, losartan, tetrathiomolybdate. I apologize if I pronounce any of your agents wrong, dear representatives from pharma and academia. And I also apologize if I don't hit all of them, because we'll be here for about 3 hours if I do. And then those that have already had negative results, as you can see, etanercept, macitexin, simtuzumab, etc. There are also some interesting pending results that are going on, including pirfenidone and vigatinib, which is a hedgehog active agent. I know this is too small to read, except for the folks in the front row, but I'm pointing you here because what it does is it actually has all these major agents listed, and I've talked about a lot of them. Again, pamrevlumab, lebrikizumab, who's an anti-IL-13. There's several anti-IL-13s that are in trial or have been in trial, as well as a whole host of other agents, including causing tolerance with collagens, and a whole host of other agents, including hedgehog inhibitors, etc. This is useful. It also summarizes what's been observed. I think this is useful for clinicians, it's useful for basic researchers, and it's useful for patients, because you might be studying a pathway and say, oh, well, why don't we try to test this? Well, maybe perhaps they've already been tested or other agents similarly have been tested. So a very, very potent resource. Now I want to talk a little bit, we'll talk about larger data sets, because I think we have to be at a time, and I think I speak on behalf of Jesse and those of us that review grants and Joe Lasky for the Pulmonary Fibrosis Foundation. I think that the mouse model is important, but we have to actually have the human data inform the mouse, not the other way around. We really have to start to use clinical data as such. And this is actually Ganesh Raghu's work that actually looks at the Medicare beneficiaries data sets and really looks that the incidence of this disease has really been very flat over this decade, 2001 to 2011. However, over that same period of time, we actually see somewhat of an increase in the prevalence. What does that mean? Does it mean that people are surviving longer? It might actually mean that we're diagnosing patients at earlier stage of the disease, because high-resolution CT scanning became quite popular over this decade. But I think that it is very useful information to sound the data sets. The other thing we have from looking at data studies is, of course, the role of aging. And there are a whole host of studies that have been synopsed here, but I think you can see that in each of these 4 studies, males are more predominant within the disease than females, and it's a disease that accumulates or manifests itself as we age. And of course, there's been a lot of work on telomeres, telomere shortening, and various enzymes of telomerase biology, et cetera, that actually has really revealed a lot about the normal aging process and the genetic basis of this disease. People ask me all the time, is IPF truly increasing over time? And I think the answer is unknown. A couple of studies show that there might be increase, and at least one study out of National Jewish shows that it's decreasing. So I don't know what to make out of that. We do know that there's a fair amount, and we're appreciating more and more cases with that. So as I came back from the weekend, this is now about 2-3 weeks ago, and went back to my clinic. I actually saw this individual, the timing was perfect. It was a 55-year-old Caucasian who actually was a mechanic. He actually repaired locomotives. And he was a remote smoker. He smoked a number of years ago. He was physically very active. He was a salt-of-the-earth working man that worked very hard for a living. Interestingly, at age 42, he had bilateral hip osteonecrosis, and he was not taking corticosteroids. He had bilateral hip arthroplasties. At age 50, he was short of breath. He had a diagnosis of megaloblastic anemia, unknown why. He had a CT scan because of the shortness of breath that showed atypical fibrosis. This was all taken care of elsewhere. His insurance denied him antifibrotic therapy, even though there wasn't a lot of ground glass. They denied it because he didn't have classic honeycombing, and he refused a biopsy. Instead, he got put on mycophenolate, and actually, unfortunately, while on that immunosuppression, he infected one of his hips. He was clearly in need of a transplant, so he actually came up to our institution to get his infected hips repaired so he could be reactivated on his transplant list. I was talking to him. I asked him, when did you start turning gray? Age 18. I looked at his CTs from home. They always commented on the lung. They never commented on the liver. The liver looked funny to me. I'm not a liver radiologist. We did another CT scan, and our radiologist called cirrhosis on the CT scan. So premature graying and cirrhosis. There's this chest ray, lots of fibrosis, kind of in the central and lower, but also the upper lung zones. CT scan cuts shows traction bronchiectasis. I think maybe at this point there's some honeycombing, but not when he was denied therapy. And of course, I believe that genetic studies hold a clue to this. We now learned over the last couple of days that we're actually dealing with genes both in the telomere biology as well as surfactant biology. There's at least 10 genes, and we learned this from Dr. Kropsky and Dr. Lloyd and Dr. Garcia. And Dr. Garcia informed us that for certain genes, there may be up to 100 different mutations. So it's quite complex. This may account for up to 20% of patients with disease. So the question is can you do anything about aging? We all get older. What about removing those senescent cells? And this is work from the Mayo Clinic, Nathan Labrasseur's group with a variety of collaborators. It was Marissa Schaefer who did the work. They obtained tissues from the Lung Tissue Research Consortium and stained for senescence, and indeed nice foci of senescent cells, much higher in IPF than in control lungs. And they looked at the gene expression profiles, and of course with IPF, the collagens and the matrix genes become more expressed over time as IPF is more severe, but also many of the senescence-associated pathways also are activated and correlated. And what they did next, I think, was quite intriguing. They went from the human data to the mouse, the bleomycin mice, and they actually showed that as you give bleomycin, the number of senescent cells increases significantly, but therapeutically you could treat it. So I'd like you to focus on the DQ, which is dasatinib quercetin, that is actually two agents that we already know about from available literature as having other activities. They might be repurposed as anti-senescent or senolytic drugs. And indeed, you see that we can really suppress the senescent cells. And if you look at this measure, which is compliance or lung stiffness, the control mice are here. Bleomycin drops the compliance. That means stiffer lungs. And you can reverse this with either the genetic approach, which is what the blue AP stands for, or the two available agents that could be used in combination. So there are a whole host of emerging therapeutics for this disease based upon clearing senescent cells, apoptosis inhibitors, dasatinib, which is a tyrosine kinase inhibitor, quercetin, which I mentioned was coupled with dasatinib, p53 inhibitors, NOX4, as was spoken about yesterday by Victor Thanakal, another target, mitochondrial targeted therapies. These senescence-associated secretory pathways might be inhibited by things like rapamycin or JAK or STAT inhibitors. And of course, there is emerging evidence, which we'll be talking about later, about the potential role of stem cell therapeutics, and I will leave that to other speakers. The bottom line is, by looking at things like the genetics and the human data, we can inform the pathways that we're addressing in cells and mice. I think this is very important. Now, as many people that know about me know that I, for a large part of my career, have studied immunocompromised host infections, patients with HIV, and fungal infections. That's my other passion other than pulmonary fibrosis. And indeed, HIV, when I started my medical training, was a lethal dead disease. And now all you have to do is know that through multi-agent treatment, look at Magic Johnson, people live a long time. We have to go on to combinatorial therapy. It's enough to say, well, let's prove that my drug is better than the other drugs. That's not the way it works. David Ho, HIV investigator, won Man of the Year for doing this in '96. But as pulmonary docs, we've been doing that for tuberculosis forever. Cancer therapeutics are almost always multi-agent. We have to move off the dime and design all of our trials to have multi-agent therapy, if nothing else, based upon having the availability of standard therapies. How have we done so far with combinatorial therapy? Well, up until recently, the news is not great. You know about the Panther data. Predase and NAC, worse than placebo. What about pirfenidone and NAC? Worse than pirfenidone alone, Jürgen Beer's study. But there is a recently released study of the combination of pirfenidone and nintedanib. And indeed, this is a tolerability study. And indeed, it showed that with nintedanib, with add-on pirfenidone, there was a manageable safety and tolerability profile. And there were some early hints that there might be some added benefits, but the study was not designed to show efficacy of the combination. It was for tolerability, but it does open the door for doing additional studies with these agents in concert. We need more agents. If you want to screen for an infection organism, you put the organism in the plate and you put a library of compounds. To that extent, that kind of broad spectrum screening has not been done. There's been a lot of targeted screening. And so actually, over the last 4 or 5 years in my lab, we've started to do some screening. We had a poster, Cassie Batzloff, Megan Gertman, and Paige Jensen did screening, and we took the Johns Hopkins repurposing library and screened by taking fibroblasts, culturing them in the presence of a variety of cytokines, mostly TGF-beta, and testing and looking for matrix generation. And actually, what we found is totally unexpected answers. We actually contrasted these to known antifibrotic agents in our test, and what you can see are classes of drugs you would have never predicted. The azole antifungal drugs showed up to have pretty significant antifibrotic activity. Recent evidence shows that they actually inhibit hedgehog pathways. Makes sense. There's already a hedgehog trial or two in that big list. But there's also indications that a variety of agents, including COX inhibitors, cyclooxygenase inhibitors, anti-inflammatory drugs, may well have benefit, including certain congeners of steroids, may have some benefit. Maybe they're beneficial in add-on therapy, not as primary therapy. And also vardenafil. And actually, interesting, of the 3 PDE5 inhibitors, vardenafil was the only one that had substantial antifibrotic activity. Of course, at the NIH, we tested sildenafil. So I think we have challenges in screening, and we need to move in the last minute. I want to move and talk about large data analytics, because it's one way to identify novel pathways, novel targets, and novel effectiveness. And in fact, we have to use those big data analytics not just to look at the biochemical, genetic pathways, but also to look at patients are faring with these medications once they're on the market. This is a nice review by a tool Butte that says that you actually can take gene expression analysis, mutational analysis, and couple these together with artificial intelligence and select new pathways for testing in the laboratory for potential drugs. Again, human data, this is human data, informs the mouse data. Very important. I'll skip that in the interest of time. Now, we're going to end up in just a second here, but we know in the real world when we take care of patients, when we do clinical trials, it's very controlled. Patients are very controlled. You see them every 3 months. They get a certain set of interventions, and you test them. In the real world, it doesn't work like that. And I think that there's a lot to learn about why some agents succeed and one agent fails. And I think, Emery, maybe you'll allude to this with NAC and TOLP. There's some patients that do well with NAC, and there's some that don't based upon their TOLP genetics. By the same token, I think we have to do post-marketing analysis. I think we need comparative effectiveness analysis of the available agents, looking at the large data set. Within the next 2 to 5 years, everybody's de-identified medical records are going to be merged and available for research. And that's already beginning. There are data sets that have hundreds of millions of patients to analyze that are available today. Last data slide or whatever, I need to also urge all of us as an IPF community, the big whammy in IPF is the acute exacerbation. You know how many clinical trials they found for acute exacerbation on NIH.gov? Maybe 1 or 2. We know that when a patient gets acute exacerbation, most people will expire within 3 to 4 months. We give people steroids at high doses. There's really very minimal, if any, data on that. There's some data on rituximab in that setting, some data on cyclosporin, but we need to do clinical trials in this entity because it matters to our patients. So, in summary, we're seeing the benefits of basic research. There's a whole host of new drug trials. There are many trials that are going to be out soon. I think that gives us all a lot of hope as patients, caregivers, investigators, scientists, and physicians. But I also urge that we need to look at this large data set analytics. And this, coupled with supercomputing, is a virtually untapped resource for new target development, biomarker discovery, but also for discovering how the agents that are already out there are behaving. It also can allow you to repurpose drugs. And again, we need to shift towards combination therapy. So what's on the horizon? We're looking east. I think we're starting to see the sunrise. We certainly don't see the bright light of day yet, but we're getting there. And this has been the best of times over the course of my career for IPF, but we have a long, long way to go. So I'll thank you. I know I ran a bit long, Jesse. I apologize.